Hello everyone, we are the small group discussion of B10 from the Bachelor of Medicine and Medical Doctor Study Program, Faculty of Medicine, Udayana University of 2022. At this moment, we will present you our student project entitled Human Blood Vessels Development. Hope you will enjoy.
Hello, my name is Komangadi Surya Mautama with the last 3-digit student number 221. Today I will tell you the background of our student project. Blood vessels are one of the important part of the human body. Blood vessels are responsible for distributing blood from heart to the rest of the body and returning it back to the heart. The blood vessels are divided into red vessels and blue vessels. The red one called arteries and the blue vessel called veins. Arteries will carry oxygen-rich blood from the heart and then circulate it through the body, while veins will carry deoxygenated blood from the body's tissue back to the heart. Okay, that's all from me. Thank you.
Hello, my name is Luha Elisa Cynthia Dewi with the last 3-digit student number of 223. Today, I will tell you more about the background of our student project. So, vascularization is a system that is very influential on the human body. The vascular system is closely related to the heart, which is often referred as cardiovascular system. The vascular system has a role as a distribution channel and also in maintaining homeostasis on the human body. From the embryogenesis phase, blood vessels are the first organ to develop. This process of blood vessels forming is known as vasculogenesis. That's all from me. Thank you.
Hello everyone, my name is Rafadir Rafadri with my last 3 digits of my student number is 244. Okay, so what is the research questions for this project? So the first one is, what is vasculogenesis? The second one is, what is fetal circulation? And then the third one, how is the mechanism of vasculogenesis? And then the fourth one, how is the mechanism of fetal circulation? And then the fifth one, what are the role of growth factors in vasculogenesis? And then the sixth one, what are the vascular abnormalities? And the last one is, what are the role of fetal shunts in fetal circulations? Maybe that's all from me, continued by my partner.
Before I continue the presentation, allow me to introduce myself. My name is Gusa Idekatana Renita with the last digit of student number 233. Here I will briefly mention the objectives that we would like to achieve in our student project. Firstly is to define vasculogenesis, to define fetal circulation, to describe the role of growth factors in vasculogenesis, to explain the mechanism of vasculogenesis, to describe the role of fetal shunts in fetal circulation, to explain the mechanism of fetal circulation, and lastly to describe vascular abnormalities. That's all from me. The next part of the presentation will be continued by my friend. Thank you.
Hello, my name is Jenarska Pramesti with the last 3-digit student number 208. So now I'm going to present about the definition of vasculogenesis. So vasculogenesis is the process of blood vessel formation which occurs by de novo production of endothelial cells and their establishment into vascular tubes throughout embryological development. Vasculogenesis is the formation of new blood vessels when there are no existing ones. The process that begins to develop the circulatory system. Angiogenesis refers to the creation of new capillaries out of the existing blood vessels. Vasculogenesis is the process through which endothelial precursor cells or angioblasts, in response to such growth factors and extracellular matrix, move and differentiate to generate new blood vessels. From different embryonic areas, human arterial and venous system developed from week 4 of embryo development. Human arterial are derived from aortic arches and dorsal aorta. That's all from me. Thank you.
Hello everyone, my name is Igusti Ayu Prasavita Hasikirana with the last 3 digits of my student ID number are 212. In this presentation, I will be explaining about definition of fetal circulation. With holding a purpose of supporting the unique physiological needs of a developing fetus, the blood circulation performed by the fetus holds a great difference from adult circulation in various ways. Vascular structures formed in early gestation provide an initial platform for gas exchange and nutrient delivery, while specialized circulatory structure that soon will be required in systemic circulation will then form much later in gestation with purpose to support fetus metabolic needs before birth. Once fetal placental circulation is sufficiently established, blood travels from the fetus to the placenta via the umbilical cord, which has two umbilical arteries and one umbilical vein. The oxygenated fetal blood is transported by the umbilical arteries and veins to the placenta, where it is turbulized, and it is returned to the fetus after being newly oxygenated and nutrient-rich. The developing fetus has specific physiological needs that may be met when delivering oxygen-rich blood throughout the body. These needs are supported by the structures that are only found in fetus. Following birth, the fetal circulation changes rapidly to make room for extrauterine life. Although fetal strips were originally used to help humans understand the fetal circulation, modern techniques like magnetic resonance imaging or MRI and ultrasound during the fetal period now offer more detailed information. The fetal circulation has differences that, if not properly formed, can cause disease in children or adults. That is all for me. Thank you.
Let me continue to the presentations, but before that, my name is Ida Ayu Pratinia Surya with the last 3 digits student number 230. Now, I will explain about roles of growth factors in vasculogenesis. The formation of blood vessels start from vasculogenesis and angiogenesis. These formations divided into two phases such as activation phase and resolution phase. For the activation phase, it triggers angiogenesis by a stimuli called growth factor. That's why growth factors play an important role in vasculogenesis. There are some growth factors that play a role in vasculogenesis, such as the first one is FGF, or we can call it fibroblast growth factor. So FGF play a role in contribute to the formations of mature vascular tissue, and it also affects mesodermal induction by giving rise to the entire embryo by the germ layer, a specific area of the epiblast. The next one is VEGF, or we can call it vascular endothelial growth factor. VEGF basically is divided into 6 based on its receptor. One of that is VEGF-A. VEGF-A binds to VEGR1 and VEGR2 receptor. VEGR2 is expressed in the first week of pregnancy or embryonic phase. VEGR2 is expressed by hemangioblasts that affect cell migration and will trigger endothelial cell differentiation. Meanwhile, VEGR1 is not actively involved because it doesn't have mutagenic effect and has a negative role on vasculogenesis. This VEGF also play a role in binding to tyrosine kinase receptor, which play important role in formation of vein. The next one is PDGF. PDGF, or we can call it platelet-derived growth factor. PDGF have a role in inducing endothelial cell proliferation. It also as an antibody that neutralizes PDGF-BB and blocking migration, and also as a paracrine route, and it also participating as endothelial and mesenchymal cell transition. The last one is TGF, or transforming growth factor. TGF play a role in inhibits the proliferation of endothelial cell and stimulate extracellular matrix repairs. And TGF play a role to initiate the growth factor response. So that's all. Thank you.
Hi everyone, my name is Komasri Bungasdani with the last 3 digit of student number is 241. I will explain about the mechanism of vasculogenesis. Vasculogenesis is the initial formation of blood vessels by the processes of differentiation, expansion, and incorporation of endothelial precursors that occur during the embryogenesis. Through several chemical mediators, the yolk sac endoderm will induce the embryonic mesoderm to form a blood island. From this blood island, blood vessels and primitive blood cell will form. Blood island is formed from hemangioblast precursor cell that arise from mesenchymal cell of this embryonic mesoderm. Hemangioblasts will later differentiate into angioblasts. Blood island that already exists will spread in the yolk sac endoderm. All of these blood islands, which later become blood vessels, will unite and connect to each other to form a circulatory system. Then, slowly, an apoptotic mechanism occurs in the central part of the blood island, which makes space appear and later will become the lamen of the blood vessel. Some angioblast cells that did not undergo apoptosis will differentiate and flatten into the endothelial lining. These endothelial cells have important roles for the body, including regulating blood transport to cells and tissues, regulating homeostasis, coagulation, defense mechanism, inflammatory response, and angiogenesis. The surrounding mesenchymal cell will also differentiate into the vascular connective tissue and smooth muscle. In postnatal and adult life, this existing blood vessel will continue to proliferate through the mechanism of angiogenesis.
Hello everyone, my name is Farel Armando Damanik with the last 3 digits of student number 243. And today I'm going to explain about the student project of SGD B10. So I'm going to explain about the roles of cardiac shunt in fetal circulation. The fetal circulation system is distinctly different from adult circulation. During pregnancy, the fetal lungs and liver are not fully developed and are bypassed by 3 shunts. This intricate system allows the fetus to receive oxygenated blood and nutrients from the placenta. It consists of the blood vessels in the placenta and the umbilical cord, which contains 2 umbilical arteries and 1 umbilical vein. Fetal circulation bypasses the lungs via a shunt known as the ductus arteriosus. The liver is also bypassed via the ductus venosus, and blood can travel from the right atrium to the left atrium via the foramen ovale. The foramen ovale enables venous blood to flow from the right atrium to the left atrium and then to the systemic circulation, while the ductus arteriosus diverts blood from the pulmonary artery to the aorta. The ductus venosus connects the umbilical vein to the inferior vena cava, bypassing the portal vein and liver. These shunts typically closes soon after birth, when the newborn starts to breathe and the lungs begin to function properly. The ductus arteriosus undergoes changes that ultimately lead to its closure, while the ductus venosus closes shortly after the umbilical cord is cut and become the ligamentum venosum. The foramen ovale also eventually closes, typically within a few weeks after birth. However, for approximately 30% of the population, the foramen ovale does not close completely, resulting in a condition known as foramen ovale patency. Fetal circulation undergoes a rapid transition after birth to accommodate extrauterine life. There are distinct differences in the fetal circulation that, if not appropriately formed, can lead to childhood or adult disease.
Hello everyone, let me introduce myself. First, my name is Komang Takas Atnyasuyana with the last 3 digits number of 224. And now I would like to explain about the mechanism of fetal circulation. The fetal circulation differs from the adult circulation in many ways to support the unique physiological needs of the developing fetus. Vascular structure formed in early pregnancy provide the initial platform for gas exchange and nutrient delivery. The specialized circulatory structure required from systemic circulation developed later in pregnancy to support the metabolic demands of the fetus before birth. The placenta connects the circulatory system of the mother and fetus. The placenta provides the growing fetus with oxygen and nutrients from the mother. It also removes metabolic waste and carbon dioxide from the fetus through blood vessels in the umbilical cord. The umbilical cord develops from the placenta and that's to the fetus. Oxygenated blood from the mother in the placenta flows through the umbilical vein, partly into the fetal hepatic circulation, but mostly into the inferior vena cava. Bypass the liver via the ductus venous, and oxygen saturation is estimated at 70 till 80%. Blood flows from the inferior vena cava through the right atrium of the heart and is sent to the left atrium. In the fetal circulation, the right atrium is under greater pressure than the left atrium. Therefore, most oxygenated blood flows from the right atrium to the left through an opening called the foramen oval, while from oxygenated blood from the inferior vena cava and from the superior vena cava mixture of deoxygenated blood becomes partly oxygenated blood in the right atrium. Once oxygenated blood reaches the left atrium, it travels through the left ventricle into the coronary artery and aorta, which branch off to supply the brain with the most oxygenated blood and the partly sandy oxygen blood from the pulmonary artery called the ductus arteriosus, with the blood supply which then flows into the systemic circulation with an estimated oxygen saturation of 60%. Partially, the oxygenated blood from the aforementioned right atrium can also pass into the right ventricle and then into the pulmonary artery due to the high perfusion resistance of the lungs. Blood flow from the pulmonary artery through the ductus arteriosus, which usually bypass the lungs, into the aorta, and the blood then enters the general circulation and the deoxygenated blood, estimated at 40% oxygen saturation, returns to the placenta via the umbilical artery for deoxygenation by the mother. That's all for me. Thank you.
Before we continue the presentation, let me introduce myself. My name is Tabita Etirnania Brusumiring with last 3 digits of student number 232. I will presentate about vascular abnormalities. Vascular abnormalities, also known as vascular anomaly, falls into two broad categories, namely vascular tumors and vascular malformation. Vascular tumors are divided into 3 categories, namely benign, locally aggressive, and malignant. In benign category, there are infantile hemangioma and congenital hemangioma. On locally aggressive has caposiform hemangioendothelioma and tufted angioma, and in malignant tumors have angiosarcoma. While for the vascular malformation is divided into 4, namely capillary malformation, venous malformation, lymphatic malformation, and arteriovenous malformation. For the specific explanation can be seen in PowerPoint. Thank you.
Hi everyone, my name is Nimade Aprilia Dwi Sisyanti with 3 last digits of student number 229. Okay, the last material is the conclusion. Blood vessels are the first organs to develop from the embryogenesis phase. The process of formation of blood vessels that occurs by de novo production of endothelial cells and their formation into vascular tubes throughout embryological development called vasculogenesis. In the fetus, the circulatory mechanism moves from the fetus to the placenta through the umbilical cord, which has 2 umbilical arteries and 1 umbilical vein. The formation process is influenced by growth factors such as fibroblast growth factors, vascular endothelial growth factors, platelet-derived growth factors, and transforming growth factors. The vascular system can also occur abnormalities which certainly interfere with the circulation process in the body, which is divided into 2 types, vascular tumors and vascular malformation. That's all about our presentation of this topic. Thank you for watching and goodbye!